good afternoon, everyone, and welcome back to the Brainy Boomer Lecture Series. We're so very happy that you've all joined us today. We would like to begin by first acknowledging that the McGill University Research Center for Studies in Aging is located and our work is done on unceded Indigenous lands. The Kanien'kehá:ka Nation is recognized as the custodians of the lands and waters on which we gather today. Tiohtià:ke, commonly known as Montreal, is historically known as a gathering place for many Indigenous peoples. Today, it is home to a diverse population of Indigenous and other peoples. We respect the continued connections with the past, present, and future, and our ongoing relationships with Indigenous and other peoples we serve within the Montreal community. In 2007, the McGill University Research Center for Studies in Aging, or the MCSH Education Committee, started the Brainy Boomer Lecture Series in order to suggest practical steps to both improve and maintain brain health, as well as to promote healthy lifestyle choices amongst the most populous generation in history. The MCSH Education Committee, which was founded in 1996, has three main objectives: identifying education needs of healthcare providers, seniors, caregivers, and the public, and to develop responses to meet some of their needs, to enhance the positive image of the aging process by addressing stereotypes and myths about aging, and finally, the dissemination of research on aging. Dr. Bernard Robert received his BA from UCLA and his PhD from McGill University. After a postdoctoral fellowship at Johns Hopkins University, he returned to McGill to take up a joint appointment in the departments of pharmacology and therapeutics and obstetrics and gynecology, where he has remained and is currently a James McGill professor. Dr. Robert's research interests focus on the aging of male reproductive system, spermatogonal stem cells, male-mediated reproductive toxicology, the mechanisms of androgen actions, and the structure, function, and regulation of the epigenetics. This research activity has resulted in over 230 journal articles, 70 book chapters, and editing and co-editing of 10 books. He's conceived and has co-edited both editions of the Handbook of Andrology. He has mentored many graduate students, 30 PhDs, 10 masters, and over 20 postdoctoral fellows, most of whom who have gone on to have successful careers in academia, industry, and government. Before continuing, we would like to remind you to please mute your microphone on Zoom, and that if you have any questions, you can write them down in the chat box on Zoom. And now I'd like to invite Dr. Robert to start his presentation.
Thank you, Caitlin, and thank you to the Center for inviting me to present this talk on a topic that you probably do not hear about very often. So should we be concerned about older dads in terms of what happens to them with respect to aging, with respect to their sperm production, with respect to changes that occur as a function of time? So the concept of andropause, the people have used the word andropause to be parallel to menopause. It's a terrible word, because in women, aging is associated with a very rapid loss in ovarian function because you deplete the eggs. In man, that doesn't happen. It's a very slow process because men produce about 1,000 sperm every heartbeat, 100 million sperm per day, whereas women produce 1 egg per month. So it's quite a different system. So instead of using the word andropause, people have used late onset hypogonadism. Probably the most common term is PADAM, partial androgen deficiency in the aging male. It's a slow process. So in fact, if we look at what happens with androgen as a function of age, we have an initial peak that occurs during fetal life that makes him male a male. Without that, an XY male will look like a female. There's another little peak after birth called mini puberty, and that marks all the tissues in the body as being male for later development. Then there's a quiescent period, and then testosterone goes up at puberty. And during the aging process, so it peaks around 20, 25, the age of our medical graduate students, and then it slowly decreases. And after about 40, the decrease is at a faster rate. So in fact, if we look at one of the best studies that has been done by the National Institute of Aging, looking at men at different age windows. So you can study people over 80 years, but you can study them over 10 years. So they've taken men of different ages and then followed them for about 10 years. And no matter where you start, you see that, in fact, if you start young, in the 20s, by the time you hit 50, 30s to 50s, you have a big fall off. If you start at 45, there's a fall off. No matter where you start, over a 10-year window, there's a decrease. So what are we concerned about when we look at aging in men? There's this gradual decrease in testosterone and in the active metabolite of testosterone called dihydrotestosterone. And that really starts moving faster after the age of 40. The testosterone circulates in the body and is bound by a protein called sex hormone binding globulin, SHBG. And that protein goes up with age. So the actual amount of free testosterone decreases even faster than the amount of total testosterone. Both steroidogenesis and sperm quality, and we'll come back to sperm quality, decrease with age, but there are also changes in libido, in muscle strength, and older men have prostate problems that are of two types, benign prostatic hyperplasia and prostate cancer. So all of these other problems associated with aging, we will not talk about for the rest of the hour. What we'll focus on is the sperm. So in fact, people say, well, old men can have kids and they're fine. So you look at people like George Lucas or Charlie Chaplin, they had kids when they were very old. However, the reality is that people are having kids at an older age because of blended families, because of long studies. They want to establish themselves. They want to have enough of an income before they start having kids. And if you look at the data, this is one sample slide, you see that, in fact, the age at which men start having children has changed. The younger men are having starting to have their family at an older age, and older men more and more are having families at an older and older age. So if we look, for example, in the 35 to 38 window, the green bars, you see that, in fact, as we move over the years, that's my pointer. There we go. As we move over the years, the green bars move to the right. And no matter what, even older one, the dark blue, also older men are having kids at more and more advanced stage. So should we be concerned about that? So I'll try to convince you that we should be. And for the rest of the hour, we will have these 5 headings. So we're going to talk about changes in semen parameters with fertility. We'll talk about chromosomal abnormalities in the sperm. We'll look at the impact of paternal age on offspring. And then I'm going to take a side out of human studies, side trip, and talk to you about the brown Norway rat as an animal model. It's a really great model to study reproductive aging. In fact, has been used for other aging studies, and we can chat a bit about that. And I'll finish with a discussion about a team grant we have that is just finishing, and I'll show you some of the results that came out of that. So, semen parameters. When you do a semen analysis, you can look at many endpoints. What happens with age is that the semen volume decreases. There's no big change in sperm concentration. So men keep on producing sperm throughout their lives pretty much at this rate of 100 million per day, no matter what. The total motility of those sperm, though, does decrease. The progressive motility, so the idea for them to be able to move fast and penetrate layers around the egg, that decreases. And there are more and more abnormal sperm in terms of their appearance as you age. So there's an effect on sperm quality for sure. There's a classical study done by some colleagues in Canada showing that based on paternal age, the odds ratio of having a pregnancy decreases. So if you look at men less than 40 and you define that as 1, by the time you hit 50, then it's only about 50% chance of being able to have a kid. So there's low quality sperm, longer time to pregnancy, and some abnormalities we'll see in a minute. So what sort of abnormalities? So this is a classical study showing at the rate of mutations. This came out from Iceland, actually. The rate of mutations in sperm as a function of age. And you can see that the mutation rate keeps on increasing. So these cells, to get 100 million cells, they have to keep on dividing, right? And to keep on dividing, you get more and more chances of having an error in the reading. And in fact, you increase mutations. So the older you are, the longer your cells have been dividing, the more chances your sperm have to have a mutation in them. In fact, that's even seen at the level of the chromosomes. So if you look at men of different ages, and you look at the structural abnormalities in the sperm, you see that the ratio goes up from basically normal up to about 3 to 4-fold higher chances of having chromosomal abnormalities, either too many or too few chromosomes, or some disjunction in the chromosomes. This is a study done in California. And what they did is that they took men of different ages, that's across the bottom on the x-axis, and they did a test called the sperm chromatin structure assay. It doesn't matter, it just looks at sperm chromatin quality. That red line that you see across the 30% is a line above which, when results from the test come above 30%, it's usually associated with infertility or problems in getting pregnant or abnormal progeny. And so what's striking is that taking men at random, you see that as you go across, there's pretty much of a linear relationship. As you age, the amount of abnormal sperm by this assay keeps on increasing. Now, that doesn't mean that a young man, you can have younger men who've got sperm that are higher, and you can have older men with sperm below the line. But by and large, there's this clear trend in terms of sperm quality, chromatin quality with age. Okay, so let's do the next, well, maybe at this point, I'll be happy to take questions along the way, by the way. So if you have any questions, you can put them in the chat box. Or if you would like to ask them directly, I'm happy to answer any questions as we go through. I don't think there's been anything so far, but we'll go on. Okay, so this is this is the press. So Time magazine had an article about fathers' ages, and is there a higher problem with kids with mental illness and other difficulties if fathers are over a certain age? And that age we'll talk about, it's either 40, 45, or 50, depending on the study. So in fact, the answer is yes. So if you look at men over, depending on the study, 40 or 45 years of age, interestingly, a lot of the disorders that have been identified are with the nervous system. So some of them are developmental disorders, achondroplasia, that's a problem with growth, and that's linked to a specific gene. But a lot of neurological disorders, increased incidence of Alzheimer's in children born to older fathers, higher incidence of autism, attention deficit disorders, bipolar incidence goes way up. But it's not just the nervous system. You have cardiac problems in children of older fathers. And the data on cancer rates are now increasing. So there were a couple of preliminary studies about 10 years ago, 15 years ago. Current data show that the incidence of cancers are going up in children when the father is over 45 years of age. So what kind of numbers? So this is one study with a very large population base. And what they looked at was bipolar disorder. The hazard ratio is huge for bipolar. ADHD and bipolar are the two that really stood out in this study. And those are not small increases. If you look at the 95% confidence interval, you can see that it is really, really high. And the cutoff in that study was 45 years. There are two other studies. This is looking at schizophrenia and looking at father's age and the incidence here. And you can see a very clear age-dependent relationship. Attention deficit disorder, ADHD, 13-fold higher risk with older fathers than with younger fathers. So it doesn't mean that every child born of a man who is over 45 years of age will have problem, but it just means that the chances of their having particularly neurological problems goes up a lot. And so it's one thing that might be really relevant to some of the populations you're looking at. This is an interesting study done in animals. We don't have enough data to do it with people yet. But so what they did in mice is to look at the grandfathers. So they're looking at the age of the grandfather, and then they're looking at the children and the grandchildren. So the F1 and the F2 generation. So the F1 would be the children, the light gray bar, the F2 would be the grandchildren. So this is a control. This is tests on social score in these animals. And you can see that if the father is older, the test on social score is much lower in the F1, but also much lower still in the F2 generation. And repetitive behavior, which is another type of typical behavior seen in autism in these animals associated with autism syndrome, you can see that that goes up in both the F1 and the F2. So it's not just the age. If the father is older, the defects that are occurring in the sperm, and we'll talk about the kind of defects, those defects are not just passed on to the children, they're also passed on to the grandchildren. All right, so to study all of this is really difficult in people because of the age and because of the length of our grants being at most 5 years. And so we really rely very much on animal models. The problem with animal models is that for most of rat studies and mouse studies, as the animals age, they have other problems other than just the reproductive problems. They usually get different cancers and different cardiovascular problems. So it's very hard to dissociate the aging problem of the reproductive system versus the aging problem caused by the other diseases in the animal models. The exception to that was identified a few years ago by the National Institute of Aging. And the brown Norway rat has become a standard animal model in aging studies. At least for the reproductive system, I don't know to what extent it's being used for neurodegenerative studies, but for the reproductive system, it's a particularly good model. And we've used it and had several grants working on that model. This has been done in collaboration with many graduate students and postdocs listed here, and with colleagues at Johns Hopkins and at McGill, listed in blue on the right. And all this has been funded by both NIH and CIHR. So what about this animal model? Well, most animals, most rats die by the age of 2 years. these animals will live to 3 to 4 years of age. So they live longer. They don't become obese. So if you have a sprig dolly rat, the standard animal model, the albino SD rat, by the time he hits 18 months, it's almost 1.5 kilos. It's almost like a mini cat. You know, they're huge. These guys level off around 1 pound, 500-600 grams. They don't become obese. So, and they don't have all the age-associated pathologies we talked about before. So it is the first rat genome also. When people want to do the genome of a rat, the default rat is the brown Norway rat. It's an inbred model, and we have very good data on the DNA sequences and all the genes associated with that. So it's a great model. We've done a lot of work with that model. We've looked at altered progeny, looked at the effects of reactive oxygen species, oxidative stress, looked at DNA damage, altered transcriptome, spermatogenesis, looked at the effects of oxidative stress, what happens with the intrinsic aging of germ stem cells, and we've looked at the epididymis and height ages. So I'm not going to go over all this by any means. I'm going to pick 3 areas, and I'll show you 1 or 2 slides for each of those areas. So if we look at altered progeny, so this was the first study actually where we took males of different ages and mated them with young females. This study was published in the late 90s and was the first demonstration that paternal age alone had an effect on progeny outcome. So what kind of effect did we have? So if we looked at pre-implantation loss, which reflects decreased fertility, we see that by 24 months, that has gone up. Post-implantation loss, which is what happens as a reflection of whether there would be more spontaneous abortions, that doesn't change. The 2 things that are more important that do change is that the weight of the progeny is reduced. And that's very consistent even at 12 months of age, you start seeing that. Most dramatically, you have an increase in postnatal death rate. So these animals are born, and the rate at which the newborn die because they can't manage goes up a lot compared to control. And so this establishes that there's something, even though the sperm are still being made, there must be something wrong with them. And we've tried to determine what that is, what is actually wrong with them. So one of the obvious questions is there DNA damage in those older males? So how do you assess damage? One way is to look at whether there's more oxidative stress, more bases of the DNA that are oxidized, and that results in the inability to read the genome. So you can do that by labeling the cells. And here we're using a brown dye, basically, that will look at the 8-oxo-DG, so one of the bases in the DNA that's oxidized. And you have 3 different cuts of pathology for the young. So this is all the testes. These are seminiferous tubules. These are the tubules where the sperm are made. These are the sperm here. And that looks pretty normal. If you look at the older male, what you see is all this brown, and the brown is in the germ cells. So the DNA in the nuclei of the germ cells. So the DNA has more oxidative stress. You can actually also take the sperm from the epididymis, so the sperm that would be coming out of the male, and you look at the young and the old, and you look at the 8-oxo-DG, the same marker, but now you use a fluorescent dye, and you see a lot more that have, so this is looking at the DNA, this is looking at the damage, the 8-oxo-DG, this is a merge of the 2, and it's clear that in the older animals, you have a lot more. In fact, you can quantify that, and you can see the fold increase in aging has gone up like crazy. So yeah, they are making sperm, all right, but the quality of the sperm, the DNA chromatin is altered. So what are the consequences of that in terms of reading that DNA? So you can do a transcriptomic analysis. That just means that you look at all the RNA that is made from the DNA, and you try to analyze what happens to all the RNA. The RNA is going to be used for making the proteins and making the cells functional. So you can look at, there are about 20,000 transcripts made in the cells. And what you can do is to look at young, can take two different cell types. So, okay, as you make the sperm, you go from a stem cell, a spermatogonia, and then these cells divide. They have to go through meiosis. They have to be half of the number of chromosomes. And then they have to reshape themselves into what looks like a spermatozoa. So packaging spermatocytes are cells undergoing meiosis, reducing the number of chromosomes. And round spermatids are the germ cells right before they start condensing all the chromatin. So what you see here, what's very clear is that those two cell types are very different from each other. So the red and the green make different transcripts. What's also very clear is that all of the young, which are at the top, separate from the old, which are at the bottom. So these are the old, these are the young. So as a function of age, in both cell types, the kind of RNA that is made is dramatically affected by aging. So what would be the consequence of that? So here we're focusing on one of the more important aspects of trying to repair DNA, right? So as cells are affected by oxidative stress or aging or chemicals or whatever, the cells have the ability to repair DNA. These are four different types of mechanism. We don't need to go into them, but four different types of mechanism by which you can repair DNA. And these are the number of genes that are, number of proteins or transcripts that are used in each of those mechanisms for repairing DNA. And what you can see is that the number altered, which is here, are very much affected by aging. So the dark gray, and one of the pathways that's most affected is that first one, the base excision pathway. The proportion of the genes affected is higher here than for any of the other pathways. And so we looked further at that pathway, and we found that in fact many of the RNAs and the proteins were affected by age. And the ability of the endogenous, the existing mechanism to repair the DNA just was not maintained. And so if there was a problem, the cells couldn't repair themselves very well. So maybe I'll stop here and see if there are any questions before we go on to the last section.
As of right now, we don't have any questions yet, Dr. Robert.
Okay,
but I have a question. Can I ask a question?
Sure, sure.
So, so there are other aspects that are associated with, I know, the health of these spermatozoids. For example, people talk about heat that they are exposed to them, and or perhaps viral infections like mumps. And so is there any are these problems somehow, I imagine that they are amplified actually with the aging process. So if somebody is somehow exposed to factors that are affecting the the reproductive system, I think this will somehow be amplified.
Exactly.
And
So this is what this slide really focuses on. In fact, your ability to repair the damage, whether it's from heat or viruses or environmental chemicals or endocrine disruptors, and as you get older, your ability to repair the damage decreases. So the cells in the body, of all the cells in the body, the ones that have the most repair mechanisms for damage are germ cells. The male germ cells are amazing. The young male germ cells can repair almost any damage you hit them with. As you get older, this ability to repair damage decreases. So you're right. That's one of the key issues.
And what are the possible, for example, medications that are normally prescribed to individuals with over 65 years old? So what are the medications that can interfere with this process?
So we'll talk about one. I don't know about medication. There's no medication to protect from the damage that I know of. What we don't know, and it's one of the points I was going to bring at the end, but we can discuss it now, is whether, in fact, things like endocrine disruptors that we're all exposed to, are the effects of those greater in men over 45 or 50 than in young men? We know that the number of sperm produced has been declining. I'm sure you've seen all these articles, and even in the lay press, that over the last 50 years, sperm production has decreased by about 50%. And those are thought to be due to environmental factors. What hasn't been done is to see the age dependency of that age decrease, of that sperm decrease. So I'm trying to collaborate with a colleague in New York who's done these studies to see whether we can see whether the age decrease is greater in men older than 40 or 45 than in men in their 20s. And those studies have not been done yet. She has the data, she hasn't analyzed them that way.
Thank you.
Anything else? Okay. So I'll move on to the last section, which deals with the STEAM grant. It was a great collaboration between Dr. Peter Chan, who is a urology colleague of ours, and people at the Genome Center, several people across the country. And so let me show you what our questions were. The first was to study the effects on telomeres, and I'll explain what telomeres are in a minute and why that matters. And the second was to start looking at some of the epigenetic marks. And I'll focus on the DNA methylation, and I'll tell you a bit about that. We've also been doing a number of epigenetic studies, a number of animal studies. I'm not going to cover the animal studies today. I just wanted to focus on some of the clinical studies. So let's talk about the first one first. What are telomeres? So at the end of chromosomes, you have repeat sequences called telomeres. Every time a cell divides, the telomere shortens. That's true for most cells in the body. The question is, what happens in sperm? Because they have, so normally cells have about 30 to 40 cell divisions. And if you look at any somatic cell anywhere in the body, and then they're subdividing. That can't happen with a sperm because they have to keep on dividing this 100 million per day for decades. So what actually happens with the telomeres? So we looked at the number of, large number of men, over 150 in the study. And we looked at men of different ages, and we divided them either in 3 age groups, that's what you're seeing on the left, or we had a continuous study analysis, same data on the right. We had 2 groups of men. We had men that were fertile, that they had had children and had semen analysis that indicated they would be fertile. And we had men who came in for the infertility clinic. They still had some sperm we could analyze, but they were infertile. And so what you see is that after the age of about 45, you have this inflection point. So the younger man and the middle-aged, no effect on telomere length. In older men, you have an increase in the telomeres. That's totally counterintuitive. For every other cell type, that should have gone down. Why you have an increase has been hypothesized to be because you're selecting the cells that are able to maintain their telomeres, and the ones that have shorter telomeres will disappear with age, and you select for those with longer telomeres. That was one finding. The second study was to really look at epigenetic marks. So before we do that, let's talk a little bit about epigenetics. So the sperm is an amazing cell in terms of its chromatin. At the top, what you see here is normal DNA, and the bases that get methylated. So you have the four bases everybody knows about, but one of the bases, the C, can get methylated. And those methylation marks are critical for determining which genes are read and which ones are not read at different points in time. And normally the DNA is organized in nucleosomes like this. So there are proteins called histones around which the DNA is wrapped. And those histones have little modifications on them as well. What happens during spermatogenesis is that most of these histones are removed and they're replaced with very basic proteins that allow for the coiling of DNA. And you retain a few histones. In humans, it's about 15%. In rodents, it's about 1 to 2%. But these histones that are retained are really important because they are the places where the genome will be first read when the sperm enters the egg. And they're the ones that activate the genome and start the zygote growth. So this toroid, these loops get compacted, and you end up with this. So you have three types of modifications. You have non-coding RNAs that are inserted in this compacted chromatin. You have the histone modifications, and you have the DNA methylation. So we're going to focus on the DNA methylation for now. So what we did is that we took young and old men, less than 40 or greater than 46, 12 control for each group, and 12 infertile for each group. And we did the complete methylome, all the bases and whole genome methylation analysis, complete the methylome, and we analyzed that. So I'm going to show you just a couple of the slides of what we found. So if you look on the right, you look at the effect of fertility. If you look at the left, you look at the effect of aging. So one of our surprises was that, in fact, fertility did not, fertile versus infertile man, you did not have a major difference in the DNA methylation pattern. You had a few that are different. So the red line would mean no change. The black line is where the data actually fell. And what you're seeing is that there's a few that are off of the red line, but not a big difference. However, with aging, you have a dramatic difference in the methylation pattern. Absolutely dramatic. Many, many thousands of sites were being affected. So where are they? So this is the same graph basically as the last one. And now what you're seeing across are the different chromosomes. And each little dot you see points to a different change in a methylation site. So, and the higher the level is, the more extensive that change is. So these are called Manhattan plots because it looks like buildings in Manhattan. And you can see that there's a lot of methylation, but they're sort of spread throughout the entire set of chromosomes. So we asked, are these the same changes as what you would see in a white blood cell, in a regular somatic cell that's aging? Are the sperm methylation change? We know that somatic cells change with age in terms of their methylation pattern. Is that the same as the sperm? So in this graph, again, you have all the chromosomes on the bottom. The red dots are the places where the somatic cells have changes in methylation pattern. These are white blood cells. The blue dots are where you have the changes in the sperm. So it's very clear that the sperm methylation pattern changes with age are totally different from what you see in somatic cells. So what are the consequences of these methylation changes? Well, one of the really interesting sidelines that we pulled out of this is just, based on these methylation changes, can you figure out how old the man is? And it's surprisingly accurate. So you can look at the methylation pattern of a male and be able to predict within 2 years what his actual age is. And that was done in an initial study with those 48 samples, and then we repeated it with another group of 10 men, and the prediction was within 2 years. So it's really precise. So the way your methylation is altered with age is pretty consistent. This slide is the one I thought you might find most interesting, and that in fact, we're looking at the sites in the genome that are either hypomethylated or hypermethylated. So you can do an analysis of all of these, and then you say, okay, I have all of these modifications, they're affecting different genes. Is there any pattern? Is there any analysis we can look at in terms of gene transcription that will give us an idea of the type of pathways, the families of pathways that are being affected? And everywhere I put a red arrow is a system that's related to the nervous system. We thought what we would have an effect on is going to be reproduction genes. And we have found almost no reproductive genes being affected with aging. What's affected are genes that dramatically affect the nervous system. And that loops us back to the clinical studies we talked about at the beginning, that in fact, the changes people are primarily seeing with aging is these ADHD, bipolar disorder, schizophrenia. So are they due to the methylation changes that are occurring in these pathways we're showing here? So that's the last of the data slide. I wanted to leave you with some of the questions, many questions we still have. And those include, what are actually the consequences of this alteration in epigenetic marks? So we've talked about telomeres and DNA methylation. There's a paper that just came out on non-coding RNAs that are changing with age. And we're working with a colleague trying to see whether we can look at histone modifications with aging. Are there, in fact, safe ways of trying to improve that sperm chromatin with age? Are there drugs that we could give? Should we be telling men to bang their sperm when they are young? Because if they plan on having a kid after 45, the chances of their kid having some issues really goes up. So that's more of an ethical, societal question. But many people are advocating the idea that before a man is 30, if he hasn't had the family size he wants, maybe he should bank his sperm. And the other question that has not been investigated, and to what extent are environmental chemicals, drugs, endocrine disruptors such as phthalates or bisphenols or flame retardants, are those going to have greater effects in older males than they do in young males? And again, there are very few people who study reproductive aging, and very few study endocrine disruptors. And so when you combine that, I don't know of any group. There's one paper that has come out indicating that in fact exposure to phthalates has much greater effects in older men than in young men. There's just one study that came out a few weeks ago. So I think people are starting to ask these questions, but we really don't have answers. And it's a wide open field for anybody who really wants to get into it. And that's all I had. I'm happy to answer any of your questions.